Hello, everybody, and thank you so much for joining us for our discussion of real-time PCR and its applications to food safety and environmental testing. Very happy that you've all been able to join us, and hope that we have a good 45 minutes to an hour with you. We are going to discuss the needs of food safety testing and environmental testing. What is it that people need, and how does real-time PCR fit into this testing paradigm? As you know, read the newspapers every day, the globalized food market is calling for much more stringent safety and quality testing. Every day we have another announcement that there is some kind of contamination in eggs. There's Salmonella in eggs. We had a Listeria outbreak in cantaloupes. We've had E. coli infections in sprouts in Europe last year. So there are many, many, many different outbreaks that are happening. They're in the newspapers and on our public conscience. And as we begin to sell food in a much more global situation, we're not going always to the local farmer, but we're going to the grocery store. Food is imported and moved around the world. The requirements for food safety testing are much more stringent and much more out there. So in food, we're looking generally at pathogen detection. In other words, is my food contaminated with pathogens such as Salmonella, Listeria, E. coli? We're also looking for the presence of genetically modified organism DNA, and that's particularly important in places with very, very strict regulations for GMOs, such as in Europe. And in looking for GMO DNA, we're looking for DNA that's been introduced into plants for particular qualities, such as conferring pesticides to the crops, such as corn with corn borer resistance, or conferring herbicide resistance. For instance, you can treat the plants with Roundup, and your plant crops will not die, but the weeds will succumb to the Roundup, so that you can get rid of weeds and get better growth. We also are looking for ingredient authentication out there. There are, especially in areas such as halal and kosher testing, where the presence of pork is strictly forbidden. We have a lot of interest in things like looking for pork, and also looking to see whether you have cheap adulterants that have been put into your food instead of using a more expensive product. In all of these, while a main requirement is that it needs to be rapid, that there's quick release of your product to the market. For instance, many products are tested when they come across a customs border, and they need to be tested immediately and released to market for sale. And the shorter the time is from test and quarantine to release, the better it is for selling the food. Also, if you are testing for pathogens, you want to be able to release your food to the market for pathogen testing immediately, or as close to immediately as possible. The test has to be specific and sensitive, that you get no false positives and false negative results, so you can be assured that the answer that you get is real. And the test also has to be easy to use, and that it can be readily and very easily adopted by laboratories. If we look at environmental testing, which is water, soil, air, the same kinds of issues are raised. Public safety demands, we want to have rapid and accurate testing for water and soil, air, and so on. A lot of this is microbial pathogen detection in water, in sewage effluent, in air, looking at, for instance, Legionella in air handling systems. Looking microbial pathogen detection might be looking for cryptosporidium in water sources and wells and things like that. And again, the testing has to be rapid so that we have limited danger to the public. It needs to be specific and sensitive, again, with no false positives or false negatives, and again, easy to use so that it can undergo straightforward adoption by laboratories. Advantages of real-time PCR in these systems are that you deliver results in approximately 3 hours. It's very quick, so it's the rapid issue. It accurately detects DNA in challenging samples, and certainly food and environmental samples can be very challenging because they're very complex. The protocols need to be simple so that you can fully automate them, and with real-time PCR this is true. It can detect specific targets without false negatives and false positives. So when you have good specificity with real-time PCR, it's a very good detection system, and it is unaffected by food processing or high contaminant contents. So real-time PCR is highly suitable, therefore, for sensitive detection of pathogens in samples of food, animal feed, water, soil, and air. And it is also highly suitable for detection for DNA detection, specifically needed for GMO detection and ingredient authentication. Okay. To get a little more specifically into the QIAGEN Maricon portfolio, which is the name for the food and environmental testing portfolio, it is an integrated system that meets laboratory demands. We have a full range of sample preparation and detection assay kits and instruments. It demonstrates top performance, sensitive, target-specific, and inhibitor-resistant chemistry. It's easy to use. We have streamlined, robust, user-friendly sample preparation and assay protocols, and it is fast, has hands-off automatable sample preparation and rapid results with low and high throughput solutions. If you have a few samples, we have a solution for you. If you have a large number of samples, we have other solutions that will be suitable for your lab. The portfolio has broad coverage. You can look at many sample types, from pathogen enrichment cultures to DNA obtained from food, from food itself. Sample types such as environmental samples, water filtration protocols, and so on. We have a great deal of success with challenging samples, and that data is available. If you're interested, we can show you more about that. Broad coverage of food and environmental pathogens. The portfolio is set with about 14 pathogens at the moment. We have a broad range of GMO and ingredient-specific assays, so the total portfolio covers approximately 35 different food environmental pathogens, GMOs, and ingredient authenticity assays. And the portfolio is standardized. We have standardized and streamlined sample preparation for each one of these various assays. We have a food preparation protocol. We have a pathogen, a bacterial pathogen protocol, and so on. And there is a single consistent protocol for all assays. So if you are interested in looking at several different pathogens in a single sample type, say Salmonella and E. coli in a single sample, you can do both of those assays in a single run, not in a single well, but in a single run. And that is a huge advantage for the protocols. This is just a diagram here of a PCR workflow for food safety testing. You start with a processed or raw food sample, and here we have a picture of food. Say this is a sausage. You can grind up the sausage and get DNA from the sausage to look for the presence of pork or whatever, some DNA in that sausage. You can also put the sausage into an enrichment culture, and you can see if you can grow out, for instance, a sausage would be something that could grow listeria. So you can put sausage into a bag of buffered peptone water, well, probably not buffered peptone water for listeria, but for an enrichment culture, enrichment medium, and from there you can get a sample that you can test for the presence of pathogen. After you get your DNA, you then put it into a Maricon assay, and you detect your presence or absence of your target. Now, another reason is why is PCR or why is there so much interest in PCR? real-time PCR as a protocol in the food and environmental segments. If you look down here on the bottom, this is the traditional culture methods. Traditional culture methods, whether they be for pathogens in food or pathogens in the environment, generally take as long as 3 to 7 days to get information because you have to do a traditional primary enrichment, a secondary enrichment, you do isolations. There are several steps, all of which require overnight incubations to finally get confirmation that a pathogen exists. So this could take as much as 3 to 7 days to get an answer. Immunoassay is quicker, but again, you generally have 2 enrichments to do. So you have to do a primary enrichment culture, a secondary enrichment, then you can do an immunoassay for detection. They're not as specific. And again, it takes a couple of days to get that information. But with real-time PCR, to get food pathogens, you generally only need to have a single enrichment, one overnight enrichment, and there's a lot of movement and work being done to get those enrichments into the 18 plus or minus hour time frame. At that point, you would take the DNA from your enrichment culture and then extract it, and then you can detect by real-time PCR. And this gives you your answers in 18 to 27 hours of your entire time. So you're really looking at less than a day, and generally somewhere in the vicinity of, say, 20, 21, 22 hours to get an answer with real-time PCR to know whether you have pathogen in your food or not. Now, if we think about PCR in general, let's move sort of to more general thought. There is traditional PCR, which is endpoint PCR. You put a primer probe into an assay and you let it incubate or cycle in a thermocycler for some amount of time, and at the end you then run a gel to see whether you had a product or not. It's fairly easy, but one of the challenges of this is that it's not terribly accurate, and certainly if you're starting to think about quantitation, which is not, if you're thinking about quantitation, certainly it's not very discriminatory, and again, it's not terribly accurate, so it would be very difficult to say there's nothing there, and one of the things that you're looking for with food and environmental pathogen detection is you really want to know the absolute presence of nothing. So real-time PCR provides you with sequence amplification, real-time detection, sequence-specific probes, and it's rapid, sensitive, and specific detection of your pathogens. And it's in real time, and it's very rapid and very sensitive. Okay, so what is the PCR reaction? PCR stands for polymerase chain reaction. It's the exponential amplification of DNA in a single tube. Now, PCR has only been around, it's really comes under one of the topic of an enabling technology for molecular biology. DNA was conceptualized, if you would, invented by Kary Mullis in, I believe, about 1983. And he won a Nobel Prize for the work about 10 years later. But it has been a technique that has really changed the way in which people can do molecular biology. Prior to that, if you wanted to make copies of a DNA molecule, you would have to make a plasmid. You would have to make your sequence. You would have to ligate it into a plasmid. You would then transfect the plasmid into a bacterium, and then you would grow up your DNA overnight. It might grow, it might not grow. Very, very difficult and time-consuming work. But with the advent of PCR, you were able to do all of this amplification in a tube, not with an overnight culture, but in a single tiny little tube in the space of just a couple of hours. So that is, really the definition of an enabling technology. Okay. A PCR reaction has the following components. You have a DNA template. It can be single or double-stranded. It has a polymerase, and these polymerases are thermostable. They can withstand temperatures of up to about 95 degrees. and that was another piece of the enabling technology, is that you could actually find these polymerases that could withstand high temperatures. Then you have DNTPs, which is the nucleotides that get added as you copy the DNA. There are forward with red and blue reverse primers. You have 2 of those. And you may or may not have probes depending on what kind of reaction you're doing. And all reagents in this reaction will be in excess, non-limiting, so that you can make as many copies of the DNA as you would like to. Okay, so the steps in a PCR reaction are to denature the template. So you heat it to 95 degrees. You anneal the primer, heat it at about 60 degrees, so you raise the temperature to 95. You denature the template, you anneal the primer by lowering the temperature to about 60 degrees, and then you make the extensions at about 72. So it's a thermal cycle. It's heat cycling, hot, cold, or hot, cooler, a little bit warmer, hot. Okay, and then you repeat this many times in each one of these cycles. Generally in a PCR reaction, you have somewhere in the vicinity of about 40 to 45 cycles to get the amount of DNA that you want. Okay, so what does this look like? Okay, we denature the template, which separates the strands. That allows access to the template for the primers, so the primers can bind. And here we have the primers binding and annealing, and they anneal. Need to lower the temperature to get the primers to actually anneal to the DNA. And then we extend the primer at 72 degrees. And again, this extension, the polymerase copies the template and makes a copy of your primer. And there you have a copy of the primer. And so you real DNA, your complete DNA. And as you get shorter and shorter DNA templates, you'll get many copies of just the sequence that you're interested in. So it would be the point between the two, the forward and the reverse primers. And then we repeat that, and we make many copies of the DNA. Because we denature it again, we anneal the primer, and it goes on. So the advantage of real-time PCR is that it's a highly sensitive and reliable method for detection and quantitation of nucleic acids, be they DNA, in this case, RNA in other cases, or perhaps a cDNA. It's based on detection of fluorescence emitted from the reporter molecule in real time. We'll go into what that looks like in the next couple of slides. The detection occurs during the accumulation of the PCR product with each cycle of amplification. So you detect the accumulation with each cycle of amplification. And as we see later, we'll accumulate fluorescence, and then we'll be able to say something about what is actually in the reaction mixture, at least in there to start with. And we monitor the PCR reaction during the early exponential phase when the first increase in the amount of PCR product is there, and that correlates to the initial amount of target template. Okay. So real-time chemistries, how do we do all this detection? There basically are two basic types or classes of chemistries that we have with real-time PCR. There are intercalating dyes. So intercalating, if you follow my pointer, and the dye will be intercalated. It will fit into the grooves or into the double helix of the DNA molecule. It's fairly nonspecific in terms of the amount of DNA that you have there, or but it will tell you how much DNA you have. So there are many situations where just knowing how much DNA is there is okay. Cyber green is probably the most widely used of the intercalating dyes, but high-resolution melt, which is not a topic of the seminar, but it's a way of learning a little bit more about your DNA. There are some HRM dyes that are also intercalating, and they go by names like LC green, Evergreen, Cyto9, and so on. But those are the intercalating dyes. However, when we're talking about real-time PCR for looking at pathogen detection, we're talking about probe-based chemistries, so that the fluorescent dye is actually on the probe and not intercalating into the DNA itself. Okay, and these probe-based chemistries generally are in the topics of a TACMAN probe. There are FRET or hybridization probes that are used for light cyclers with Roche, molecular beacons, linear probes. There are a number of different ones. For the purposes of our discussion, we're going to be talking about TACMAN probes. TACMAN probes are also called hydrolysis-based probes, and how this works is that you have a probe that has a fluorophore on one end, and the other end of the probe has a quencher. So as long as the fluorophore and the quencher are both on the probe, you get no fluorescence in your solution. You start with your forward PCR primer and your reverse PCR primer being set down on the template. And the probe, of course, is looking at the specific region that you're interested in probing, in looking for, identifying. So as the polymerization takes place, you bind, you extend the molecule, and as all of this gets closer to where the probe has been laid down on the molecule, once it gets there, the newly made DNA and the polymerase will actually knock the probe off. And when the probe is displaced, the fluorophore now goes into solution. It's cleaved from the fluorophore is cleaved from the probe, and it enters the solution. And because it's not close to the quencher anymore, your fluorescence is no longer quenched, and you get fluorescence in your solution. Okay, and then ultimately you get PCR products, and you get fluorescence in your solution, and the rest of the probe is because the TAC has a 5' 3' exonuclease activity, the probe will be chewed up, and all you have left then is this fluorescence. The fluorescence is because there's one probe per every molecule. The fluorescence is concentration. The amount of intensity of fluorescence is then related to the amount of initial template that you had in your assay. One caveat is that each amplicon needs to have a sequence-specific probe, but that's just you need to have sequence-specific probes. You can't work with a generalized probe. Okay, so how does a real-time PCR cycler work? So we started off, we know that we're going to make fluorescence, and we need to look at the fluorescence, be able to detect that fluorescence in some kind of a machine. So you have a thermal cycler which does the 95 degrees, the 60 degrees, the 72, so it cycles the heat, and with each one of these cycles you generate a fluorescent molecule. How do you see that? How do you detect that? So there's an excitation source. The molecule is excited with some kind of light at some particular wavelength. The excitation source can be a halogen lamp, it can be an LED, it can be an argon laser. This light then goes through focusing filters, lenses, and mirrors that then lead it to the detection device, and the detection device can be something like a CCD camera, a photomultiplier tube, which is what the rotor gene has, or a photodiode. There are all these different detection devices. But basically, a thermocycler is some kind of an instrument that heats and cools in a cyclical manner. It has a light, an excitation source, and a detection device that allows you to see what the accumulation of fluorescence from your reaction. So the rotor gene Q, which is QIAGEN's thermal cycler, is not a plate-based cycler. You'd be very familiar with these 96-well plates. Instead, we have a rotary system. We have a reaction chamber here, and into the reaction chamber we have our thermocycling, our disc that contains our samples. So this is a rotor, and there's a number of samples. It can be anywhere from 36 to 100 samples in that disc. The tubes are then in a rotor, and because the rotor is spinning, it spins past the optics, and the speed is about 400 RPM, so each one of these tubes is interrogated with each wavelength that you're looking at every 400 times a minute, basically. There is a spindle motor assembly at the bottom that turns the rotor. We have an LED light source, so we have something that will excite, so the LED is the light source assembly for the rotor gene. And we have a PMT detector, photomultiplier tube. So there's a lens to focus the light that's being made, detection filters to give you the right color that you're looking for. So you, with each cycle, you have an LED light that's being focused on your tube. You then look through the lens and you detect the light with every cycle. Okay. Now, one of the things that's very interesting about the rotor gene is that instead of having a plate-based heating system, which can have hot spots and cool spots because of the Peltier cooling system, heating and cooling systems that are there, have a rotor which brings hot air in from the room and heats the air in the chamber and then cools down with pulling cool air in from the room. So it works like this. There's a chamber vent that seals it to contain the air. You turn on the heater, and the centrifugal fan drives the air around the chamber. And because we have holes in the rotor disc, or the rotors themselves, it will allow the airflow to happen in the chamber. Here's a picture of a rotor with holes in it to allow free airflow. And then if we want to cool, we switch off the heating element, turn on the fan, which will bring the cool air in from outside, and we drive the cool air into the chamber, and we expel the hot air by opening the vents. And this happens every time we're working on with every cycle. So you heat, you cool it down, you heat it up a little bit more, heat it up more, and then cool it down for the cycling. Okay, so in real-time PCR, we have a number of terms that you will hear, and what are these terms? What do they mean? So it's a little bit of real-time PCR terminology. We have baseline, threshold, CT, crossing point, or no template control. So let's look at each one of these terms. A baseline, as I said, we accumulate fluorescence in sort of a baseline fashion, and these are the initial cycles in which there's little change in fluorescent signal. So usually cycles 3 to 15, this thermal cycling, we get no accumulation of fluorescence, and at some point we begin to accumulate fluorescence here. So this area right here is called the baseline. Now, once we've accumulated enough fluorescence, we begin to generate more fluorescence, and this is the amplification comes above the baseline. So here's the baseline, and then at some point we begin to accumulate fluorescence, and that point at which it goes above the baseline is called the threshold. And you will see this in terminology in a lot of your real-time PCR. And then that point where the baseline crosses this threshold is called the threshold cycle. And this is probably the most used terminology for real-time PCR. What's your CT? And this CT can move. It's an indication of how much, of a couple of things. It's an indication of how much fluorescence you have there, because the fluorescence is accumulating, and you're accumulating enough and eventually become quite exponential. And then also this threshold cycle can move depending on whether you have an inhibited reaction. So if a reaction is inhibited, and you compare two reactions, same DNA, same reaction chemistry, but there's something different in one tube than in the other, you can get an inhibition, and what will happen is that CT value in an inhibited sample will move over to the right. So left is more DNA, right is less DNA, but less DNA can also be an inhibited reaction, and that will become important, or the importance that will become evident here in a moment. And then one other term that you see is no template control. That's a no nucleic acid template in the reaction. So you need to have one of these to make sure that you don't have any contamination. PCRs are very easy to get cross-contamination. You can get little bits of DNA that appear because you need to be extremely careful in your laboratory not to get extraneous amounts of DNA into a tube. It can come from your hands, it can come from the air, it can come from another tube. So these are things. So you control for the presence of gross cross-contamination by making sure that you have a no template control in every one of your assays, so that you should get a flat baseline saying that you have no contamination there. Okay? So critical factors for a successful assay. What do you need to do in order to have a really successful assay? Well, your DNA or RNA sample preparation has to be extremely good. You have to get good template quality. It needs to be clean as possible. So you need to choose appropriate sample preparation kits or reagents that will remove as many inhibitors as possible, because inhibitors can compromise your PCR. As we said a moment ago, if you have inhibitors, you can actually move the CT, which is the indication of sort of goodness of your reaction, can be moved far to the right with an inhibition. Assay design has to be specific, efficient, and good chemistry. When you design your assay, you need to consider your throughput and cost per result, and you need to use thoroughly validated assays, whether that be an assay, for instance, a Salmonella assay that has been given PTM status by AOAC or given validation status by AFNOR. These are validations that are done by external labs to make sure that the assay works well. If you have an assay that is not going to receive that kind of validation, because there are only certain ones that do, you will have to yourself validate the assay in your lab, and that becomes very important. So there are validation things that you might want to do. If you're running your PCR, you need to choose high-quality reagents, so good primers, good probes, good master mix, and also make sure that these master mixes are tolerant to inhibitors in your assays. And then you want to have data analysis tools that are user-friendly and have good data analysis modules. So the effective inhibitors, just sort of to show you, here's an endpoint PCR, which I think shows very nicely what happens when you add inhibitors to PCR reactions. So we go from no inhibitor to more inhibitor, because it's here. And there were 3 assays. So here's one with no inhibitor, a little bit with SDS, you have a little bit of inhibition, and at the high level, you don't see a reaction at all. You see nothing. And so this is through a number of chemicals that could possibly contaminate a PCR reaction. As you can see here, in every case, when you have higher levels of the inhibitor, you get an inhibition, either full inhibition or a very markedly reduced amount of DNA that's there. And then to move this into the Maricon assay, what we did, knowing that inhibitor tolerance was very important, we did a series of studies where we looked for at all of our master mixes. Zucchigen has a large number of different master mixes to see which one had the best tolerance for inhibitors. And the inhibitor models that we used here were things that you would find in various food preparation assays, just sort of as a model, but chemical inhibition here. So we used CTAB, sodium chloride, isopropyl alcohol, ethanol. The blue are the Maricon assays, and the red or purple are from an assay from another supplier. Now, if you have a tolerance for an inhibitor, what you should see is if you have an assay with the inhibitor and one without the inhibitor, you should see very, very, very negligible amounts of change in the CT. So you want to see basically no effect of the CT change. And for all of these, there is essentially, there's certainly less than a CT cycle difference between the inhibited and between the assays with the inhibitor and without the inhibitor. However, if you take another multiplex assay, another assay from another supplier, and then do the same experiment. So we have plus inhibitor and no inhibitor. You get a CT change. And here with CTAG, we're seeing about 6 CT difference. So it's moved from the first point, let's call it, maybe it was 28. I don't know what the number was, but let's just, for example, the first one was 28, and then we have a CT change of 6. That moves us into 34. And the CT of 34 is getting into the point where it becomes more and more difficult. I mean, this to actually see your DNA. Sodium chloride moves it 11 CTs to the right, which means basically you won't be able to see your product unless you have a really, really strong sample. And isopropanol, you still have a CT change to the right of a couple of CT units, and ethanol about 5 CT units. So all of these are really point out the importance of having a good multiplex master mix. And now you can talk a little bit about internal controls, because one way you can control for the presence of an inhibitor is by using an internal control. And all of our Maricon assays use a green fluorescence for the specific pathogen. And then there's also in that same reaction an internal control that has a yellow fluorescence. So you have a green and a yellow fluorescence in the tube. And you can tell whether you have a valid result, because if you have the amplification of the internal control takes place, then it's valid. If you have a lot of inhibitors in that sample and you get no or reduced amount of internal control amplification, you can say that result is not going to be valid. So it's a nice way of checking to make sure that you had good amplification. There are some assays that we have, especially lab-developed assays, where you can put the internal control at a higher concentration into your sample and use that to follow both extraction and amplification. It's not going to tell you which one didn't work, but it will tell you if you have a valid result. So, again, the workflows for our food and environmental testing, we have disruption, and this disruption can be lysis. It can, depending on your sample. For a bacterial culture, it will be lysis of the culture. For a food sample, it will be taking the food and homogenizing it as a first step in the purification. For a water sample, this first step is going to be some kind of collection of bacteria or pathogen on a filter generally, or some kind of, let's call it a filter medium. And then you need to disrupt that. You need to somehow lyse the cell walls. Things like Clostridium that are very difficult to lyse, or Clostridium spores, or something that you have big challenges in the disruption. Then you get you purify your DNA, set up your reaction. So we have solutions for manual or automated purification. We have solutions for either manual here or automated setup of your PCR, and then you do target detection, and detection in the Maricon system is done with the rotor gene. Here's a lab. This is a sample preparation lab. So here we have, this is a QIAsymphony instrument on the table. Here's a rotor gene. So we take our samples, put them into the QIAsymphony, and do our food prep that way. Here we have sort of an example of, here's a food prep, but you can say any kind of early preparation. We then use a suitable DNA extraction kit on that first step. So Maricon DNA bacteria or bacteria plus kit for overnight pathogen cultures, DNA the Maricon food kit, which is a CTAB-based kit for food stuffs to get DNA from food for either ingredient authentication or GMO. And then for water and environmental samples, we have kits such as the QIAamp UCP pathogen mini kit. So we make the DNA, and at the end of the sample prep, we have DNA molecules. The DNA molecule is then added to a suitable Maricon assay or lab-developed assay. and these Maricon assays are very simple because you basically make a master mix and you add DNA to the master mix and put it then into a PCR instrument, and we get results. There's a little picture of results on a computer. We have assays for a broad range of specific pathogens, genetically modified organisms and ingredients, and we have a lot of kits suitable for lab-developed assays, which are very important in the food and in the environmental arena. Just a quick look at the software for real-time PCR setup on the Rotor-Gene. Essentially, you have you just say, I want to run 95 degrees for 15 seconds, and then you set up your cycling parameters here. If you wanted to move them for warmer, change the temperature, change the time, it's very easy to just grab onto the bars and grab and pull. Set up 40 cycles, and then you're ready to go. It's a very straightforward setup of your PCR reaction. So what did the results look like? These are some examples of results from a number of our different arenas here. On the top figure, we're looking at Salmonella detection in peanut butter. Peanut butter is a pretty difficult matrix. And here we have 1 to 100 dilutions, up to 1 to 100,000 dilutions. So there are 4 different dilution ranges here, and you get nice sensitive detection of the Salmonella in the peanut butter. The next figure here on the bottom is detecting pig DNA. Again, we talked a little bit about the desires of halal and kosher food manufacturers to make sure that there's no pig present in your foodstuff. For instance, a lot of gelatin is made from pork rather than from some other pork. Well, made from pork, and you don't always know that the gelatin is pork-based, and so you can use this to detect pork and gelatin, the presence of cheap meat in more what's deemed to be expensive meat, but it's actually cheap, and so on. So it's a nice way to look at ingredient authentication. And again, we're detecting down to 3 copies of pig DNA in this particular assay. For water testing applications, we have a large number of we have many citations here for water testing applications available. If you go onto the website and look for environmental assays, you can see a large number of citations for different water testing applications that have been published using QIAGEN techniques. All of these things are very flexible. They can be used on any cycler. They can be used on a Rotor-Gene, but if you have other cyclers in your lab, you can use the QIAGEN protocols for that as well. And they're convenient setup. No need to optimize the reaction. Because of the high tolerance to inhibitors, you need to do far less optimization to generate your validated assays. On the left, we have a graph showing detection of norovirus, and this is in vitro transcribed norovirus using the QuantiFast pathogen RT-PCR plus IC internal control kit. So there is a very nice kit that can be used. This was on the Rotor-Gene, and here we have both duplex and singleplex. So this is showing that there's no adverse effect of having a duplex reaction in the kit. And the duplex is that we have the norovirus probes plus the internal control in the same reaction tube. And here we see a dilution series for the norovirus. And this is just looking at the internal control for all of the samples, and they're all quite similar. These are examples here on the bottom of pathogen nucleic acid detected with the Quantitek probe and RT-PCR kits. So it's a wide variety of different environmental pathogens. We have a Maricon kit for Legionella species and Legionella pneumophilia, and then there's quite a bit of work that's been done on Cryptosporidium with QIAGEN techniques. And I believe there's also, if you look at the QIAGEN web, there was webinar presented by a customer fairly recently that talked about Cryptosporidium. This is some validation data that for Legionella pneumophila, and you can see that this is basically what we did was we set up a dilution series with Legionella, so we're starting with 10 to the 7th copies all the way down to 10 copies, looking at how well we can detect it, and then this is just the internal control up here, showing that the internal control worked and there was no inhibition of the assay. And again, same kind of data for Salmonella species detection. And we have all of the assay targets gave us a green fluorescent channel, i.e. a Salmonella-specific detection. And here's our internal control, which was detected in the yellow fluorescent channel. And a little bit more data from food samples. This is chocolate with coffee filling. Both of these are foodstuffs that have a large number of inhibitory substances, methylxanthines, things like that can inhibit PCR. And what you see is very nice dilution curve showing you that you get DNA and you get detection of the DNA even down to very few copies. Here's the milk chocolate dilution series as well, and internal controls for both of those. So what we provide in real-time PCR support is easy-to-use systems that are fast to learn and to implement with basic training in every lab, large choice of validated workflows depending on throughput and regulation, access to R&D and application labs for specific development requests. We have we have R&D people and apps labs all over the world who can help you. We have technical support available in English and in local languages, and that tech support is available 24 hours a day for 6 days a week. We have field service engineers who are familiar with routine testing lab requirements. And last but certainly not least, QIAGEN has extensive experience in molecular testing, having done been active in the field of molecular testing, both from sample preparation and assay development side and instrumentation development, of course, for about 25 years. So in summary, real-time PCR is a rapid, sensitive, and reliable method for the detection of pathogens in food and environmental samples. Real-time PCR is highly suitable for specific DNA detection needed for GMO detection and ingredient authentication, and QIAGEN is here to support you in your food safety and environmental testing work. So thank you all for attending our webinar.